Good morning, everyone. I'm Colin O'Connor, Custom Content Editor at Genome Web, and I'll be your moderator today. The title of today's webinar is Novel Expansion and Repertoire Profiling Technologies for Immuno-Oncology and Autoimmunity. This webinar is sponsored by Cellecta. Our speakers today are Alex Chenchik, President and Chief Scientific Officer at Cellecta, and April Mueller, MD Ph.D. candidate in the Medical Scientist Training Program at Albert Einstein College of Medicine. You can type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. And if you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. Before we get started, we have a poll question from the sponsor. If everyone could select the response that best applies to you. Do you have experience in your lab using TCR or BCR-seq for immune repertoire profiling? A, yes, we have experience running these in-house. B, yes, we have sent samples out for a contract lab to run. C, no, we have no experience, but I'm considering using this technique within the next year. And D, no, we have no experience, and I'm here to learn more. And we'll give just a few seconds for everyone to answer. Okay, I think we can take a look at the results. So D, no, we have no experience, but here to learn more with almost 80%. That's most of you. And C, no, we have no experience, but considering using the technique within the next year, about 20%. So with that, I will hand it over to Alex Chenchik.
Introduction. We are going today to talk about immune receptors. And immune receptors, if you study in the school, it's the most complex genetic system created by nature. If you look at the slide, I would like to remind you that B-cell receptors, B-cell receptors, T-cell receptors, and antibodies, which are soluble form of B-cell receptors, are generated, organized very similar way, with CDR3 regions shown in circle, recognize foreign antigens. If you think from biological point of view, it's a universal diagnostic system created by nature in order to recognize any foreign antigen or pathological conditions or molecules in your body. And complexity of this system is just enormous. It's at least 100 million of different immune receptors are constantly present in our body, and they're organized in generated by genetic recombination from V, D, J, and C regions, rather random way, creating this enormous complexity. The practical question, if you're talking about diagnostic system, how to use this system in order just to identify any pathological conditions or disease in our human bodies. Problem here, why it cannot find broad application, because we don't have really direct link between immune receptor sequence and specific antigens which they recognize. If we solve this problem, in this case, it will be an outstanding really system which can be used in order to diagnose practically most of human diseases. From practical point of view, how to study this immune system? The question is enormous complexity. If you think, what does it mean 100 million of different immune receptors? It's at least 1,000-fold more than the number of the human genes. If you are talking about to apply the same tools as what you use for gene expression profiling, you can see where's the problem, because can we analyze 1,000-fold more complexity than really the number of the human genes? And so today we are going to, in order to facilitate these studies, we developed several technologies like illustrated on the next slides. And first, it's adaptive immune receptor profiling assay, which is allowed to do it in bulk profiling of TCR and BCR clonotypes. And we use clonotypes with the name of specific CDR3 sequence, which corresponds specific clone of B cells or T cell. We also going to talk slightly about single cell RSA for T and B cells sorted in 96-well play, which allow to get full-length chain pairing information for immune receptors, which are necessary for understanding their function and study their properties. And top markers, about 30 markers for T and B cells. And we also slightly we're going to talk about genome-wide expression profiling, which allow to do it bulk expression profiling of all protein coding genes. And all these technologies are designed based on the multiplex PCR. So that's why they are rather similar in concept, how you're going to use them. And we are going to discuss the application of this technology for screening of activated immune receptors with peptides or vaccines, and how to validate this type of the interesting clone types what you identify in the course of these studies using artificial reporter system based on JURCAT and K562 cells. As introduction to the technology, here is the example of the assay what we designed for adaptive immune receptor or RSA profiling assay. And what you can see here is structure of immune receptors. On the top you can see you can use this assay not to amplify from RNA, like shown at the top, or from DNA, like shown at the bottom. CDR3 region shown here in the center, which is, and it can be amplified using flanking primers, reverse primers, or forward primers. In this case, you amplify CDR3 region. What great in this technology, what we developed, you can amplify all 7 chains for all TCR and BCR genes. So it can be alpha, beta, gamma, and delta for T cell receptors and HKL for B cell receptors. So you can start from RNA or DNA, and RNA provide you more sensitivity. DNA give you more quantitative measure of copy number of the T cells or B cells, which has specific CDR3 sequence. You can amplify only CDR3, like shown here on this slide, using combination of internal primers, or you can amplify if you use FR1 and combination with C primer, you can amplify full-length region, which includes all CDR1, 2, and 3 regions of V gene of the immune receptors. What great in this technology, you can not only use RNA or DNA as starting material, you can directly use cell lysate and not to amplify all these molecules. Quantitative analysis facilitated of using validator barcode. And validator barcode is similar concept to universal unique molecule identifier, which allow you to correct number of different mutations introduced in the PCR on next generation sequencing step. And data analysis are integrated with IMXR and platform software package, which is dominating platform right now for immune repertoire profiling. In terms of protocol, here's the illustration of another assay what we developed. It's genome-wide expression profiling for 19,000 protein coding genes. And here number of the primers is significantly higher. We have here 19,000 reverse primers, like shown here on this slide, 19,000 for the forward primers, which is designed to amplify small region between 100 to 250 base pair or from open reading frame of most human protein coding genes. Sensitivity is higher than RNA-seq technology. And again, it's more simple protocol than RNA-seq because it can start from the total RNA without ribosomal RNA or globin depletion. And the last technology what we are going to introduce today, it's single cell analysis technology. It's some sort of the simple technology which allow you, if you just use, for example, expansion of or stimulation of T cells stimulated by proteins or B cells stimulated by any proteins, you can sort activated cells using a cell sorter in 96-well plate and perform both single cell profiling of T cells or B cell receptors and the same time markers. And these cells can be sorted based on the expression of activation markers or staining, for example, with MHC peptide tetramer or any labeled protein. As a result, you can get a sequence of the T cell and B cell receptors and expression of 30 marker genes. And in order not to be boring about talk about technology, let's try to talk about how the data analysis. First question is rather simple. Can we quantitate immune repertoire profiling data? Is it possible or not? If you try to run this experiment, for example, take purified RNA from PBMC or whole blood samples and run assay what I described, you generate approximately 100,000 clonotypes or different CDR3 sequences as illustrated here on this slide. What good here, you have rather balanced number of TCR clonotypes, approximately 100,000, and BCR clonotypes identified using this assay. TRD and TRG, it's gamma and delta chains of T cell receptor, it's very rare, so that's why their number is low. But the question is, first question when you try to run this assay, are these data reproducible or not? And simple answer, it's not. The data are not reproducible. If you try to repeat this experiment in triplicates and compare a set of CDR3 sequences what you identify, you will see that at least 99% of the sequences are 99,000 from 100,000 are amplified only from single molecule, like shown here on the y-axis. You see his starting copy of mRNA molecules used for amplification of these immune receptors. Definitely, if you have one molecule in your RNA sample, it's random choice. You amplify or not amplify. In order to get statistically reliable data, you need to have minimum 10 molecules. So this group of the genes, approximately 1,000, it's really allow you really to do quantitative analysis. And next slide is illustrate. Definitely, if you concentrate it on the top abundant clonotypes which present in your biological sample, in this case, you can get reproducible data, is illustrated here on this slide for the RNA or for DNA. For DNA, definitely it's require more DNA, which is from practical point of view, it's more complex because not always you have several micrograms of genomic DNA. So RNA will give you opportunity to really to generate good reproducible quantitative data using significantly small amount of the RNA between 10 to 100 nanogram if you are talking about blood or other lymphocyte-rich samples. The next question is rather practical. Okay, we identify all these 100,000 clonotypes. What we are going to do with these guys? Definitely the number one problem in this type of the studies, how to identify clonotypes which activated by, for example, different disease conditions or drug treatment or, for example, vaccination. And in order just to give you example how it works, this technology, I give you example of vaccination studies. In these vaccination studies, we just collect samples, whole blood samples from triple vaccine, tetanus, diphtheria, and pneumonia, and Shingrix vaccine. We collect multiple time points. It's so classical longitudinal studies where you compare samples before vaccination and after vaccination and go to look at the expression profiling data and identify activated clonotypes. If you try to run driver map genome-wide expression profiling assay, which allow to measure protein coding region in 19,000 protein coding genes, we can see for vaccination 1, after 2 days of vaccination, we can see activation of innate immunity pathways, like shown here. And approximately at 1 week, we have a bunch of the genes which belongs adaptive immune responses. If we look at the same, we run in the same samples immune repertoire profiling assay, we generate data like illustrated here on this slide. If you look at the right portion of this slide, which show IgH, so it's heavy chain of immunoglobulin or B cell receptors. You can see if we amplify from DNA, like shown here, at approximately 1 week after vaccination, for both vaccination 1 and vaccination 2, we have strong activation, expansion of the clonotypes for maybe 50, between 20 to 100 clonotypes, which belongs to the B cells, which probably we can speculate it's really proliferation of these clonotypes, which differentiated in plasma cells and start to secrete immunoglobulin G M molecules. The same data can be generated using RNA assay, but you can see immediately on this slide the sensitivity of detection of clonotypes is dramatically more sensitive than using genomic DNA, which is rather easy to explain because plasma cells have activation at least 100-fold of BCR gene in the course of activation. If you look at the T cell receptor, which are to sections on the left slide, section of the slide, you can see if you use DNA, we don't see dramatic activation of expansion of any T cells, but we still can see activation of T cells at the level of the transcription. So which means probably that T cells and B cells work cooperatively in this type of the immune responses. Based on this result, we speculate that really it's a rather good idea to run both rDNA and rRNA and normalize data to each other. In this case, you have chance to identify activated clone types by comparison these two data sets. And maybe for longitudinal studies, which I described, it's not really very relevant because you have multiple time points, but you have only one single sample. In this case, it's maybe good strategies to detect activated clone types. And next section, okay, we identify activated clone types. And typical classical example, similar example, in order to do it right now, it's a lot of studies in order to develop immune cell-based therapies. And next presentation will be describe these approaches in really big details. But what people are doing in these studies, they identify candidate peptides, which, for example, associated with the cancer. And it can be multiple strategies, including experimental, like mass spec identification of immune-specific, cancer-specific peptides eluted from the cancer cells, or prediction of the cancer neoantigens based on the mutation, changes in splicing, or any other biological process. After prediction, you need to screen these peptides, and the best strategy is to screen against PBMC or from normal or disease samples. And as a result, you can develop cancer-specific vaccine or cell therapies. And here's the classical example how it's done, this experiment. So if you take PBMC from healthy or disease donor sample and treat them with candidate peptides, which you predicted, you, after some stimulation for which can be 10 days or 2 weeks or 3 weeks, you really identify expansion of the clones like shown here on the bottom of the slide. So you see here it's in control, it's practically no expansion, and after 10 days we can see some fraction of the clone types which are expanded. After, usually in this experiment, we are doing restimulation at approximately 10 days or 2 weeks after primary stimulation, and really to identify activated T cells by sorting, for example, in 96-well plate, or using single cell 10x genomics assay. So we just illustrate that we use a combination of rDNA and RNA assay in order to detect activated and proliferating clone types, and the really single cell assay in order to identify these activated clone types after second stimulation. And you can see after second stimulation, if you compare results, and this one is classical bubble plot, where the size of each spot corresponds to the size of clone type, number of the T cells for each specific CDR3 sequence. We can see if we stimulate normal PBMC cells from normal donor with CMV peptide, we can see bunch of activated clone types like shown here on the bottom slide. Important to mention that data are not perfect, because if we just take control DMSO control, we also can see some sort of activated clone types. The question is how to identify correct clone types from these studies. And in order to do it, really, we believe that it's the best strategy is to look at the overlap between different data sets. It can be, for example, rDNA or rRNA, say, or sorting of the cells based on the secretion of activation marker CD137. Overlap between these different data sets can give you really set of candidates of T cell receptors which correspond, which activated by specific peptide. But when you identify this candidate T cell receptor, still it's only beginning of the story. You need still to validate this result. And what we find, we look at many technologies developed right now for validation of interaction between T cell receptors and candidate peptides. And we believe that really one of the best strategies to use artificial reporter cell lines, Jurkat reporter cell lines, in combination with K562 cells. And in this system, which was developed already more than 20 years ago by many researchers, including Rosenberg from NIH, what we are doing here, we just take Jurkat cells like shown here on the left side, and we convert these cells in reporter cell line, where we clone and fat reporters under control of transcriptional activation promoter, which express GFP if T cell receptor is activated. We knock out in these cells endogenous T cell receptor and introduce CD8 molecules in order to convert Jurkat cells to CD8 cells. So if you clone in this cell, you can clone in these cells individual construct like shown here on the top of this slide, or library of T cell receptors in order to create library of T cell receptors in Jurkat reporter cells. In K562, you can clone any peptide in the context of HLA complex. It can be fusion, like shown here on the top of the slides, which is peptide, beta-2 microglobulin, or HLA, or it can be mini gene, which allow you to express on the surface of K562 MHC complex, which will be exposed peptide. If T cell receptor recognize peptide, TCR signaling pathway is activated and Jurkat reporter cells start to be fluorescent. So this system can be used in order to screening individual constructs or libraries against each other. And what we find the best way to screen this type of the interaction is to use fluorospot microwell array developed by company Celldome with the name CloneXplore. And this one's great system. It's based on the using of the microwells, like shown here on the top of the slide. So it's standard 96 well plate. In each well, you have 3,000 microwells. This microwell shown here in details on the left portion of the slide, and you can see here what great in this system. You can monitor in time how many cells you have in each well. How many antigen presenting cells or T cells you download in these microwells. After that, you can look at the proliferation during the time when they co-incubated to each other. And great, each, like shown here on the right slide, section of the slide, you can see microwells are coated by antibodies against cytokines. So if any cells activated in this type of the interactions, cytokines secreted and can be immediately detected by staining with the antibodies against these cytokines. So you can see fluorescent signal. In the case of K562 in Jurkat cells, it's more simple because Jurkat cells start to be green and we can immediately monitor if they are activated by peptides. So in order to show how the system works, we developed library like shown here for 120 peptides, which is cancer-associated peptides and neoantigens. We clone them in K562 cells and incubate with the CMV TCR constructs and co-culture these two cell lines in CloneXplore. And as we just definitely selected GFP positive cells, and you can see here on the right side of this slide, we identify practically in all cases CMV peptide, which illustrate power of this technology to screen different T cell receptors and peptide candidates against each other. So we believe that really functional discovery of functional TCR epitope interactions based on the stimulation of PBMC with peptides in vitro, it's really can be effectively performed by analysis of proliferating clone types and in combination of full-length chain pairing information using combination of bulk RSA and single-cell RSA. It's also important to really identify activated clone types based on the expression of activation markers or cytokine secretions. But what is great, it's really in any case you need to validate these studies using cytokine secretion clonotype assay, microwell fluorospot assay, which I described in the previous section of my presentation. And all these technologies are available at Cellecta if you're interested to use these technologies for your studies. And a summary of my discussion, we discussed development of adaptive immune or air profiling assay, which is bulk profiling assay for all 7 chains. We discuss development of single-cell assay, which is cost-effective analysis of phenotype and full-length immune receptor parent chain information in T and B cells sorted in 96-cell plate, and with screening strategies for screening of TCR activated by synthetic peptides, vaccines, and how to validate these functional TCR antigen interactions. Thank you very much for your attention, and we are looking to answer your question, and I give turn to the next presenter.
So much. My name is April. I'm a medical student at Albert Einstein College of Medicine, and I recently defended my thesis, which I'll be giving you a short snapshot of the work that I did here today. So today my talk is called TCR Repertoire Profiling Reveals Clonality of Immunostat Primed CD8 T cells. And before I begin, I'd just like to say I don't have any financial or non-financial relationships with the products or services described in this presentation. So today I'm going to be telling you a story about my thesis work that was recently published in the Journal of Virology over the summer, and I highly encourage you all to check it out if you're interested in this work. We have a much more in-depth analysis of what I'll be talking about, so I'm just going to give you pretty much a surface level of what we looked at and how we use the technology described to inform our results. So I'd like to begin by telling you a little bit about the rise in popularity of adoptive cell transfer as a form of immunotherapy that uses CD8 T cells. So ACT, adoptive cell transfer, or adoptive cell therapy, is mainly used to treat leukemia right now, but it's rapidly expanding to treat other diseases such as solid tumors, viral-associated tumors, and autoimmune diseases. Indeed, the global T cell therapy market was estimated to be about $2.8 billion in 2022 and is expected to reach $32 billion by 2030. So this is a huge expanding market. So there's a lot of interest in this field and how to make these therapies better. So with the approval of Tecartus, Yescarta, and Kymriah, a lot of different entities have shifted their business models from the development of small molecule and protein-based treatments to adoptive cell therapies. And there are indeed numerous clinical trials currently ongoing that evaluate both CAR T cells and TCR therapies for a variety of targets. As you can see here, CD19 is one of the most common targets that used in a wide variety of leukemias. And for TCR therapies, you may recognize some of these antigens, such as HPV, a viral-associated antigen, MART1, a melanoma-associated antigen, and so on. So with all that in mind, how do clinicians or laboratory researchers actually produce these T cells for adoptive cell transfer? So CD8 T cells are first harvested from the patient's blood through a process called leukapheresis. Then they're genetically modified using lentivirals or retroviral agents to express either a chimeric T cell receptor, a CAR T cell, or a TCR against their antigen of interest. These cells are then expanded ex vivo in a process called T cell expansion or rapid expansion proliferation, and then they're then infused back into the patient to treat their cancer, their viral-associated cancer, or their autoimmune disease. Alternatively to genetic engineering, you can actually generate a pool of polyclonal CTLs through activation by an antigen-presenting cell, such as a dendritic cell. So this method using dendritic cells has a lot of benefits, and generating these polyclonal T cells has a lot of benefits, such as the ability to target intracellular proteins. If you're familiar with CAR T cells, you know that they're required to generate, or they're required to target a surface cell protein. So by using TCRs, you can target intracellular proteins, which is really important, especially for viral-associated cancers and some cancers that don't actually express those viral or those cancer-associated antigens on their surface. Additionally, they have high sensitivity. These internal antigens are very sensitive for cancer sometimes. These neoantigens are very rare, and so the ability to target these enables a very high sensitivity. And you don't actually have to identify a TCR when you're using this. If you can expand a polyclonal system, you don't need to identify the gene of each one, although it can be important to identify those genes as well if you want to convert it over to a genetic engineering method. Polyclonal TCRs are also very important for a broad immunogenic profile. And so if you have a cancer and you have an immunotherapy that only targets one antigen, there's that potential for antigen escape if you're relying on only one TCR to do the job. If you have a wide variety of TCRs that are able to target the antigen, that may be more resistant to immune escape. And finally, no genetic engineering is required, which removes any sort of risks such as malignant transformation of these cells after you infuse them back into the patient. And so this all seems well and good, but a lot of the times this method uses APCs from the patient themselves, such as dendritic cells. And this results in a very high complexity. As I personally know, it's not easy to make dendritic cells in the lab. I spent a lot of time doing it. It's not a fun thing. It's very time consuming. There's a lot of logistical complexities. It can be very costly. And actually you get a lot of variability in your result, which results in inconsistent cell qualities. As you can see here, there's a lot of different cytokines needed. It's a very long process, can take up to a week, and you actually get a very low yield at the end of the day. So overall, this results in the high cost and variability, which isn't great for adoptive cell therapies, which you want something very consistent. On the other hand, artificial antigen-presenting cells may circumvent these issues. Artificial antigen-presenting cells, also called AAPCs, provide an off-the-shelf system that can be tailored to each patient without the need to generate fresh dendritic cells for each therapy. Additionally, these modular systems enable fine-tuning of the signals given to CD8 T cells, which potentially can create more potent and longer-lasting CTLs. So we love this consistent quality and this modular system. But what exactly are artificial APCs? What do those look like? So artificial APCs can come in many different flavors. First, they can be either polyclonal or antigen-specific. Polyclonal activators use general activation signals, such as CD3, that activates all T cells, both CD4 and CD8 T cells. And this is very commonly used in things like CAR T cells, where you want to generate a wide variety of T cells, so it activates everything. On the other hand, antigen-specific activators use a peptide MHC1 complex, which will only activate T cells that recognize the target that you've loaded onto that MHC, such as a peptide. Additionally, they can be either cellular or acellular. As you can imagine, cellular APCs are derived from cells. So they can use insect cells, mouse fibroblasts, the K562 cell line, as we previously mentioned, and these are genetically engineered to express these activation signals. So cellular and acellular can also be polyclonal and antigen specific. So you kind of pick one of each half. However, they must be removed from the infusion product, or else the cells themselves can create an immunogenic response, which can be harmful to the patient, especially the non-human cells, such as the insect cells and the mouse fibroblasts. On the other hand, we have the acellular artificial APCs, which use some sort of matrix that is bound to the activation signal, such as latex beads, polymers, gold nanoparticles, or pure protein. But again, a majority of these products also need to be removed from the infusion product because they can generate an immune response in themselves. However, I want to focus your attention on one specific artificial antigen-presenting cell called Immunostat, which was the focus of my thesis. Immunostat was developed by the Almo and Goldstein labs at Albert Einstein College of Medicine, where I did my PhD, in collaboration with Q Biopharma. Immunostat is a completely acellular, antigen-specific, and entirely protein-based artificial antigen-presenting cell that is potentially infusible to patients, as it does not contain any synthetic matrix or non-human components. Therefore, it's entirely biocompatible. IST is built from two protein chains. It has a light chain and a heavy chain. The light chain contains the beta-2M chain from MHC class 1, bound to the antigen of interest, so that's modifiable. You can essentially put in any antigen that you want. And the heavy chain contains an Fc backbone, similar to an antibody, bound to MHC class 1. And this is then bound to a costimulatory molecule, which can also take many forms, such as anti-CD28, 4-1BB, or even a cytokine such as IL-2. So this product is actually currently in clinical trials, and previous reports from our lab have shown that the IST framework containing an anti-CD28 domain enabled antigen-specific growth of CMV or HIV-reactive memory CD8 T cells. So taking patients that have CMV or have HIV, we were able to expand in vivo these cells in a mouse model that were able to prevent viral reemergence. Additionally, collaborators have reported the expansion of HPV-16 specific CD8 T cells, which is from HPV, the E7 antigen, using an affinity attenuated IL-2 co-modulator. And this is the product that's currently in clinical trials for head and neck squamous cell carcinoma due to HPV. So all of this work that I've described such far has been using pre-existing memory CD8 T cells. So we're relying on the patient's own immune system to initiate that response, and then we amplify it. However, my thesis had a different idea. What if we can specifically use IST to prime naive CD8 T cells? Could we preemptively target conserved antigens or escape mutants? And targeted naive CD8 T cells would actually enable us to generate de novo immune responses. So we don't need to rely on the patient's own immune system at the beginning. We can infuse these patients with reactive TCR cells that can target a viral antigen or a cancer antigen without them ever seeing it before. So this is essentially a potential vaccine platform. So that brings me to the hypothesis of today, where we wondered if Immunostat could provide sufficient TCR engagement and co-stimulatory signals to drive activation and differentiation of naive CD8 T cells into functional effectors. So let's get into some data. But before we begin, I want to talk about the IST construct that we started using. So this used the MART1 antigen, which is, I mentioned previously, a melon A protein. So it's a melanocyte differentiation antigen that's commonly found in both normal melanocytes and melanoma cells. It's a very common antigen that's currently used in adoptive cell transfer. And it's actually important for us here because it has a unique property in that it has a very high precursor frequency. And what that means is in a normal naive component or a naive compartment, there's a lot of cells that are able to recognize MART1. In fact, it's approximately 1 in 7,500 naive cells in any healthy person that are able to recognize this MART1 antigen. So this is really important for us because it kind of hedged things in our favor. If we're able to get expansion here, that was a really good sign because there's a lot of available cells for expansion. So 2 constructs were made utilizing this antigen. First, use an anti-CD28 scFv, which is the very classic co-stimulatory marker required for naive activation, and one that did not have a co-stimulatory domain. So this was our negative control that we termed modless. So to go over how we actually stimulated these cells, so first I have a graphic depiction of what we did. First, PBMCs from healthy donors are thawed, and then naive CD8 T cells are isolated using immunomagnetic sorting. We then culture these naive cells with either our IST constructs or dendritic cells that we made in the lab as our positive control. We then culture in media with supplemented cytokines for many days and taking multiple time points throughout this time. And then at day 25, we gather our samples and we run a variety of different analyses on them, including phenotypic analysis, intracellular staining for cytokines, and cytotoxicity assays, as well as immunoprofiling. So now we can actually get into some data. So first, I see here we have a FACS plot showing day 0. And so you can see here we stained with tetramers to detect the antigen specificity of these cells. So in this donor, we have 0.16% of the starting population is able to recognize MART1. So that's our baseline that we're starting from, and we're hoping culturing with either dendritic cells or IST can expand this number. And you can see this is exactly what happened. So here we have representative flow plots at each time point after co-culture with either peptide-loaded dendritic cells on the top row, anti-CD28 MART1 IST in the middle row, and the modless MART1 IST on the bottom. And you can see how both dendritic cells and the anti-CD28 IST expanded a significant amount of MART1 specific population, 45% and 47%. So huge expansion. We were really excited to see this. And it also highlights the requirement for anti-CD28 in this expansion in that MART1 IST that was modless doesn't have any expansion at any time point. And this was very consistent with the multiple donors that we tested. So next I wanted to see if these cells were actually functional. Sure, we can expand them, but can they kill their target? So in order to assess the cytotoxicity of these samples, I ran a europium release assay, which treats peptide-loaded T2 target cells with a molecule that fluoresces upon lysis, enabling us to measure killing through measuring levels of fluorescence. So here you can see I have the different samples that I tested. In black, we have untreated samples, so cells that have not seen any sort of antigen-presenting cell. In pink, I have cells that saw dendritic cells, and in green, I have cells that saw the IST. And as you can appreciate, the IST-treated cells have a really high levels of cytotoxicity at almost all the EDT ratios tested. And specifically, they had higher cytotoxicity compared to the dendritic cell samples, which is a really interesting finding. And additionally, it is a very specific killing in that cells that were cultured with T2 cells without the peptide in the open marks didn't have killing. So it's a very specific killing and very potent killing that the cells that were treated with IST showed. So it was really exciting for us to see. Now, before I get into the next data set, I just want to highlight that antigen recognition is not a one-to-one occurrence. One antigen can not only be recognized by one TCR. In fact, one antigen can be recognized by many unique TCRs. And this is a concept called polyclonality, which I'm sure you're all familiar with. And so we wanted to assess if there's a difference in polyclonality between cells stimulated with dendritic cells versus cells stimulated with the IST construct. So I know Dr. Chenchik kind of went over what this process looks like, but just for our purposes, we assessed TCR diversity through the process called RNA-seq profiling. So we used the RNA version of this process. So this technique uses RNA from flow cytometry sorted tetramer positive T cells. So we sorted our activated cells through tetramer. We isolated these cells and we purified the RNA and sent them up to Cellecta for analysis. And so the resulting library was then sequenced via next generation sequencing, and the final data package was processed using R to pull out and quantify the alpha and beta TCR chains, as well as their corresponding CDR3 regions. And I do want to note, because this is a bulk RNA method, I couldn't conclusively make claims about the alpha and beta pairs, but it did enable us to look at the diversity as a whole. So first, we looked at total occupied repertoire space. And to imagine what this means, imagine 10 cells that each have a different TCR. Each of these clones, which are different colors, occupy equivalent repertoire space of approximately 10%. So this indicates a high diversity. After simulation, you can now see that there's two clones, the green and the yellow, that are occupying 90% of the repertoire space. So overall, a lower number of clones occupying a higher repertoire space indicates less diversity. I hope that makes sense. So here's the first data that we have, which looks at millions of clones simultaneously. So in dark red represents the top 10 most common clones. First, you can appreciate that in dark blue in the unstimulated samples, so our baseline level of diversity before any stimulation, you have a high occupation of low copy number repertoire cells. So there's a lot of different diversity going on, a lot of different cells, each with a different TCR. Versus after activation with dendritic cells or IST, you see there's an expansion in these higher clonotype indices. Specifically, the dark red here in IST is taking up more than 25% of the total repertoire space. So that means in these cells, the top 10 clones are taking up a majority of that space. Versus in the dendritic cells, you can see the top 10 is taking up approximately 15%, and then the next 100 is taking up another 50%, et cetera. So overall, IST shows more occupied space in these top 10 clones, indicating that there's lower total diversity in the IST treated samples. So next, we wanted to break down what these top 10 clones are actually looking like. So these pie charts represent the top 10 clones broken down into the unique identified CDR3 regions. As you can appreciate, the top 10 clones occupy 16% of the total repertoire space in dendritic cell treated samples, but 83% in IST treated samples. And this was consistent between analysis of the alpha and the beta chains, which was very important because they should be pairing technically, so they should be relatively equivalent. Furthermore, you can see here the most common clone in IST is actually taking up about 25% of that total repertoire space. So during the FACS isolation, you can imagine we isolated 100 million cells. 250,000 of those are actually a single clone that's recognizing the MAR1 antigen, which was really fascinating to find. So lastly, we looked at if there was any overlap in the CDR3 usage between all the significantly upregulated chains. And overlap here it implies that dendritic cells and IST are priming the same TCR chain. There's a match there. So we found 2 overlapping chains for the TRA and 1 overlapping chain for the TRB. And although this might be an artifact for low sequencing depth, it may also indicate that dendritic cells and IST are actually expanding non-overlapping clones, which again was a really interesting finding. But altogether, these data show that IST treatment primes a more focused TCR repertoire than dendritic cell treatment. So that's all well and great for MART1, but the lab that I worked in was actually an HIV lab. So we wanted to apply these findings to another clinically relevant antigen. So we looked at SL9, which is an HIV-derived epitope from the GAG protein. There's reactivity towards SL9 commonly found in chronic HIV infection, but not so much during acute HIV infection. And indeed, there's no previous success in generating responses against this antigen using artificial APC methods. So this represented the gap in the literature that I wanted to focus on during my PhD. One thing that complicates this antigen is it actually has a very low precursor frequency. So if you remember for MART1, it was about 1 in 7,500. Here it's 1 in 500,000. So clones that are able to recognize this antigen are much rarer. So we were kind of worried that maybe this wouldn't work just because we couldn't find these clones in our samples. However, we actually found they did expand, which was really, really interesting and really exciting for us. So here you can see that SL9-loaded dendritic cells actually didn't expand SL9 at all, versus anti-CD28 SL9 immunostat got up to 18% of the total culture after 25 days, which was really exciting to see because, again, this hasn't been done before. We were able to expand HIV-reactive clones in an HIV-naive donor. So essentially, we were able to generate this anti-HIV response in somebody that has never seen the HIV protein before. So this essentially was a potential vaccine platform that may provide protection against HIV in people that are healthy, that don't have HIV. But again, we wanted to do a further analysis on these cells. However, I do want to mention, because the dendritic cells didn't expand, we couldn't do the same comparisons. So instead, we just looked at the HIV responsive clones and kind of compared them to the unstimulated samples instead of the comparison of untreated versus dendritic cell treated versus IST treated. So similar to before, we first looked at SL9-loaded T2 cells, looked at their cytotoxicity, and you can see here that there was significant cytotoxicity at multiple E to T ratios, especially compared to the untreated samples. And indeed, it was also significantly killing over the non-peptide loaded samples. So it was not only significant killing over background, but it was significant killing over the untreated cells and T2 cells that weren't loaded with peptide, which was really exciting to see because it showed that these cells were actually able to target SL9 loaded cells. But additionally, that's not really a physiologic system, especially for viral cells. So we wanted to see if SL9 stimulated cells by IST could actually suppress HIV. So here we did an HIV luciferase suppression assay. So what this is the HIV genome for this specific HIV strain includes luciferase, and that when HIV is infecting a cell, it expresses luciferase and you have a high RLU, so high relative light units. And so when you kill those cells, you actually get a decrease. So that I just wanted to point that out because it's kind of the opposite as the previous system. So here, because the IST-treated samples were able to kill HIV-infected cells, we have a significant decrease in the relative light units, indicating that these cells were killing HIV-infected cells, naturally infected cells, not specifically SL9-loaded cells, which was also really exciting for us to see because it showed that naive cells from a healthy donor that was treated with IST was able to kill HIV-infected cells, which bodes really well for potential future work using this in real patients. But again, we wanted to look at the human profiling. So how does the diversity of these cells look like? Interestingly, these cells were much less diverse than MART1. However, that kind of made sense to us when we talked about precursor frequency. The initial cell samples that we used, so the initial cultures had about, well, they did have exactly 100 million cells in that sample. And for the MART1 antigen, 1 in 7,500 of 1 million cells is about 60 to 70 cells per well. So you have a lot of potential cells that were able to be activated. Versus the SL9, 1 in 500,000, there's only approximately 2 cells per well that were able to expand. And that's kind of what we saw here. The top 10 sequences for these 2 samples, which we looked at 2 different donors instead of comparing to SL9 or comparing to dendritic cells. So here we have something called donor 55 and donor 86. And in both cases, the unstimulated samples, again, we have a lot of this dark blue, there's a lot of variety, whereas the stimulated samples, almost 100% of the repertoire is taken up by the top 10 clones. And this itself didn't really tell us, well, how many clones are actually in this top 10? So we looked at the pie charts again, and as you can see, there's 1 clone that takes up over 50% of the whole entire repertoire. And then you have 2, 3, 4 other clones that are taking up a smaller amount of the repertoire. But overall, this was able to generate a polyclonal response as well, which was very exciting to see because a lot of SL9-based immunotherapies now uses just one TCR clone, and there's a lot of issues with immune escape. So what we thought is if we could generate a polyclonal response, that may actually help prevent immune escape. So in conclusion, IST is a novel artificial antigen presenting cell that enables naive antigen-specific priming for both cancer and viral antigens. The resulting CTLs from these experiments show that IST is able to enable differentiated, polyclonal, polyfunctional, and cytotoxic CD8 T cells. And therefore, IST is a potential new immunotherapeutic agent that can specifically be a potential vaccine platform, enabling patients to preventively target either cancer antigens or viral antigens before they even see them. And again, I'd just like to highlight the paper that I recently published on this work. It dives a little deeper into what these cells look like. We look at epigenetics, we look at memory marker assessment, we look at cytokine expression, and there's a lot of really interesting findings there that would have loved to highlight here, but just due to time, I had to kind of take the most important facts here to show with you guys today. And I'd just like to acknowledge everybody that helped me with these experiments, and specifically Dr. Pariso at Cellecta, who helped me analyze and understand all of this immunoprofiling data. And with that, I'd be happy to take any questions.
Great, thank you, April. As a reminder to the audience, if you have a question, you can type it into the Q&A box in the control panel. We'd like to ask attendees to take a moment after the webinar has ended to take our exit survey and give us your feedback. The first question we have here, Alex, I think this came in during your portion. It appears that AirRNA needs cDNA as a substrate and therefore doesn't work well on fixed tissue. Do you offer a version that can be used on fixed suspension cells or even FFPE tissue?
Okay, so it's rather common problem. A lot of people has FFPE samples, and it's really, they don't have anything else, and they collected maybe for the last 5-10 years and no way to go back and collect more. So FFPE, it's a create problem in honestly in any immune repertoire profiling assay, because both DNA and RNA usually damaged by fixation. And we compare rRNA and rDNA assays in order to amplify this immune repertoire profiling. The first comment is about rRNA assay. Don't use cDNA as a starting material. It's used, unfortunately, I did not really make it clear, but it's use RNA. And first step is hybridization step between reverse gene-specific primers and RNA. We just take, it can be cell lysate, or it can be FFP RNA purified, or and you hybridize it in high salt buffer set of reverse gene-specific primers with the RNA. After you purify this complex and run follow-up assay. So we start from the RNA. If you start from RNA from damaged samples like FFP, the number one rule, if RNA is completely degraded to nucleotides, you cannot amplify anything. So RNA should have at least fragments, at least 300 base pairs in order to be amplified. In some cases it's work, in some cases it doesn't work for FFP samples. DNA, in spite of significantly less sensitivity, about 10-20 fold, it's probably more robust in order to work with the FFP samples based on our experience. At least we have very good results with some FFP samples using RNA, but DNA is more robust. And a second rule, it's desirable that FFP samples will be reasonably fresh. If they're extremely old and stored, for example, for 10 years, chances for success dramatically reduced. Okay.
Got it. Next question we have here is for April. Did you also compare propagation after IST and DC? Is the oligoclonality after DC due to proliferation of T cell with lower affinity, which IST cannot stimulate?
Yeah, that's a great question. So we did look at propagation in terms of quantifying total cell numbers, and DC did enable higher quantity of T cells, and we do think that is due to the wider variety of T cells clones that it is stimulating. And we think that this is because dendritic cells, as you know, have a lot of different molecules on the surface, a lot of adhesion molecules, a lot of different co-stimulatory molecules, versus IST, which is a very pared down system. So we think that wide variety of co-stimulation, cytokines they might be expressing, et cetera, is probably why they're enabling a wider polyclonality. So future experiments that we do have is trying to genetically modify dendritic cells to kind of pare those down as well to see if we can equivocate it, as well as using IST with different co-stimulatory molecules such as 4-1BB, or even making IST with different cytokine molecules as well. And we do have one in the works that contains IL-15, and it actually also does a really great job, but we haven't gotten quite to the immunoprofiling stage for that one yet.
Alex, can you briefly outline what you think are the key differences or benefits of TCR or BCR-seq using Cellecta's approach versus other approaches, such as smart technology or adaptive service?
So let's start from adaptive. Adaptive is one of the most popular platform, and it's based on the DNA. And we just offer a variety of these services, including DNA and RNA. So it's create a whole toolbox where you can select what you want to amplify, full-length region, or, for example, adaptive technologies, it cannot amplify B-cell receptors or full-length receptor because they use DNA. So you're losing information about complete structure of B-cell receptors for follow-up validation studies. In spite that it's really very limited, it's great technology. A lot of people use it because it's rather quantitative and allowed to quantitate accurately copy number of the T cells which are expanded or present in the biological samples. In terms of smart, it's really great technology which I actually developed, I don't know, more than 25 years ago at Clontech, and it's really great technology, but it's generate big amplicon, and it's a problem in deciphering of this information. And it cannot be used for DNA, so that's why it's really multiplex PCR. It looks like it's most better strategy, especially not only in the terms of, because it's focused on the functional isoforms, because a lot of isoforms of T cell or B cell receptors are not functional. So it's amplify only functional isoforms. And on the top of this, it has significantly more sensitivity multiplex PCR than smart technology. If you try to amplify array cells from tumor samples, for example, multiplex PCR can easily amplify one cell in 1,000 of tumor cells. A smart technology cannot do it because it's not enough specificity of this reaction in comparison with the multiplex PCR technology. It's more complex that I try to cover some key elements, but I can talk about it for 30 minutes, but probably it will be too much.
Yeah, I think we have time for one more question here. This one's for April. Did you check CD28 expression at the end of your expansion, and are IST and DC stimulated T cells phenotypically comparable?
Yeah, so that's a really good question. We didn't check CD28 because something that we found is when we culture cells with IST that contain CD28, it actually blocks that receptor from being stained. So it wasn't a reliable way to look at the CD8 T cells. We did use other markers of expansion, such as CD69, and I do highlight in that paper what those results look like. And in terms of phenotypics, they're actually very different. Dendritic cell stimulated cells have a much lower differentiation profile versus the IST treated cells have a more terminally differentiated profile, and that is highlighted by the epigenetic studies that we did. And that doesn't bode great for IST for use in ACT. But again, we are working on future IST products that might be able to kind of recapitulate that lower differentiation profile. It also could be a question of we haven't restimulated with the IST, so they just get that one dose. CD28 is a very potent activator. So there's a lot of different variables that went into the system, and we're kind of tweaking and playing around with those currently in the lab.
Great. Well, that brings us to the top of the hour here. So that's all the time we have for today. We'd like to thank April Mueller and Alex Chenchik and our sponsor, Cellecta. If we didn't get to your question, we will provide them to the sponsor to follow up. As a reminder, please look out for the survey after you log out to provide your feedback if you missed any part of the webinar or if you'd like to listen to it again. An archived version will be emailed to all attendees. Thank you, everyone, for joining us.
Thank you.